Work, but really, really exciting. Um, Hopefully, guys, you like it. The paper was accepted in Science Immunology a few weeks ago. So you have it over there, but I'm more than happy to share it to anyone afterwards. So I'm going to start talking about this thing of meningeal Tregs. And just to put everyone in the same plane, what are the two words that make this title? And let's start with what are T regulatory cells? I'm going to do also. Um, can I move it? Okay. So T regulatory cells are a subtype of T cell that are characterized by the expression of CD4s. T cells can be CD4s or CD8s. These are CD4 positive T cells that express FOXP3. It's a transcription factor that is really important to know because it's exclusively expressed on regulatory T cells. There's no cell in the body that expresses this transcription factor. So this allows us for a lot of specificity and cell manipulation on this subtype. Other molecules that are highly enriched on this subtype is CD25, for example, and is the high affinity receptor for IL-2. And I'm going to ask you just to remind this because it will be important for the last part of the talk. So Tregs has been canonically known for control both sides of the immune system as immune regulators. They can control the response and fine-tune it and shape it of the adaptive branch, T cells, B cells, plasma cells, as well as innate cells such as dendritic cells, macrophages, and NK cells, controlling cytokine production, antigen presentation, and phagocytosis and function of innate cells. But I'm really lucky to be in a lab that pioneered the concept of tissue cells, especially tissue Tregs, and they have a non-immune function. What this means is that Tregs, they see every tissue in our body, and they will adapt to that tissue specifically. They will adapt the transcriptomic machinery and then the proteins to perform the best function in that tissue. And they not only talk to the immune cell, they also talk to the stroma and the stem cells. Indeed, they have a higher tendency to cluster next to the stem cells in the tissues. And in those, they regulate maintenance, proliferation, even differentiation of these cells. And as I mentioned before, they will adapt. As we each of us adapt our message depending the audience, despite being the same message, Tregs will do the same to its tissue to have a better fine-tuning efficiency on the regulation of the tissue they are sitting on. So they control the immune system, they crosstalk with the stroma, and they adapt to where they are, to which stroma they're talking to. Now, what are the meninges? The meninges is one of the three layers that are called brain borders. The brain only has a one resident immune cell, that is microglia, a subtype of macrophages. And around them, there is a huge constellation of immune cells. And from less immune populated to more immune populated, we have the perivascular space, choroid plexus, where the cerebrospinal fluid is produced, this liquid that acts as an airbag for the brain and is mostly myeloid cells, and the meninges, that is composed by three layers. From closer to the brain parenchyma to closer to the skull, we have the pia mater and the arachnoid. These two are known as leptomeninges. There's barely any immune cell. There is only one subtype of resident macrophage called Virchow macrophage, and then the dura mater. In this cell, there is lymphatics. There is also where the blood is drained from the brain and the cerebrospinal fluid is drained from the brain. And you find the whole constellation of immune cells here. So are there Tregs in the meninges? They are. There is around 15 to 20% of the CD4 positive T cells. And before starting with any data here, I'm going to make three remarks. First of all data that I'm showing here must have been perfused. So the blood had been removed from any potential blood contamination. On top of that, we did intravascular labeling of CD45 positive cells, so a marker for pan-immunocyte marker, and we addressed the contamination, which is around 10% after perfusion of blood-derived cells. Second thing is all these numbers that I'm going to show is an underrepresentation of the real numbers. Due to technical feasibility, I can only take the dura mater that is from the ears on the top, so only the top part of the skull. So the meninges that below the brain and in the spinal cord, I'm not analyzing them. So this is an underestimation of the real numbers. And last thing, yes, this one is regarding terminology. Any cell that is CD4 positive, FOXP3 negative, I will be referring them as T-cons, T-conventional cells. So going back, we found these Tregs in the dura mater of the meninges, around 15 to 20%, and they encompass around 125 cells. When I say meninges, I should say dura because, yes, as a sanity check, we check the leptomeninges and the choroid plexus. and we didn't find barely any T regulatory cells. So all the Tregs are in the dura mater. Where they are located, to that we did imaging, and we assessed where this FOXP3 and the reported GFP was presented, and we see that they are next to this structure that you can see here in white that are called the sinuses. Here, the sinuses is where the cephalospinal fluid and the blood coming from the brain is drained, and also the lymphatics goes along here. So they are hotspot for antigen presentation. And they are forming clusters with these MHC class 2 positive cells. You see that the purple are Tregs, the red is MHC class 2, so antigen presentation, and purple tends to be always next to these MHC class 2 cells. On top of that, Tregs, and you can see from this image already, they're always close to the sinuses, and here's the quantification. So the T regulatory cells tend to form clusters with MHC class 2 positive cells that in fact they are CDC2, conventional dendritic cell type 2, and this was validated by flow and by histology, and they tend to be around the sinuses. Outside them, it's kind of immune disorder for T cells at least. So after knowing the Tregs are next to whom, we assess some common physiological variations such as age, sex, and location. And I'm more than happy to discuss about some of them in the questions time, but I'm going to show you about the age because it's important too for the latest part of the talk and the follow-ups that we are doing with this. We see that Tregs enter into meninges really early on, around 3-5 days post-birth. They remain in the meninges, and after around 1 year, they start increasing in percentage and the same in numbers. We sort these cells and we sequence, and I just want to show you that as the cells age, as the organism age, there is a loss on division capacity, an increase on pathway related to proteostasis, a failure to maintain the proteostasis, the normal protein components on these Tregs. And this may be a reason why they are increasing, because they're trying to compensate the function that they cannot perform as well as before. So are these T regulatory cells homogeneous or they are heterogeneous? For that, we did single-cell RNA-seq, and we saw that there's at least 3 clusters of meningeal Tregs. One that we call it quiescent. This is a really interesting cluster because we have found in every tissue that we have checked, there's always this cluster present. Then we have a small cluster that's called T follicular regulatory cell-like. These cells are in charge of maintaining B cells in the germinal centers, controlling the germinal center size, and preventing autoantibody production. And then we found the major cluster in these meninges that are the Th1-like cluster. These cells are characterized by the expression of the transcription factor TBET, and they are optimized to suppress type 1 immune responses, interferon responses, specifically interferon gamma responses. So, of course, we always, that we do any transcriptional assay, we always validate by protein, and we did our protein validation is single cell protein validation, also known as flow cytometry. And we find with the markers CXCR5 and PD1 double positive, and TBET positive here, and a combination of all markers that are in the paper, this quiescent cluster. So now, great. We know that there are Tregs in the meninges. We know that where they are, next to whom, even how heterogeneous they are, but do they matter? To that, we use the specificity that I was mentioning before of FOXP3, and we have a FOXP3 diphtheria toxin receptor mice. The diphtheria toxin receptor in the mice is not expressed in any part of the organism. So when we give diphtheria toxin, we can deplete specifically the cell that is expressing the receptor, in our case, T regulatory cells. So here we did one thing, that is, we did a really short time course. Prolonged Treg depletion, become autoimmunity. To avoid that, we check what is the farthest that we can wait. Do not see splenic, and in the spleen we are using as a kind of as a reporter for systemic inflammation or systemic status of the organism. We didn't see any changes in the spleen, but we start seeing changes in the meninges. So that's why we did D5. And when we give this, we see that the depletion works perfectly. And before that, we did 2 other approaches to be sure that we are having a localized meningeal effect. First of all, early time course. We did 0, 12, 24, and 72 hours post the first DT dose. And we compare meninges to spleen. And we compare early activation markers. And we saw that only in the meninges, you start seeing changes happening as early as 12 hours post the first DT injection, whereas in the spleen, we don't see anything past the 72 hours. On top of that, we did intracisterna magna injection. Cisterna magna is a region in the brain that is where the cephalospinal fluid is accumulated next to the cerebellum. So you can inject it there, and everything will be drained to the meninges afterwards, as I mentioned before. And for that, we use anti-CD25, this marker that I also mentioned before that was highly enriched on regulatory T cells. And we and others have validated for many years that you can deplete regulatory T cells with that. We did this local injection and recapitulated exactly the same as I'm going to show you with the DTR, which was an expansion of T cells and a drop on B cells. And this goes along again with those classes I was showing before, TFR and TH1. To further interrogate these, first, are those T cells expanding locally? Or are those T cells going places that they should not be or they weren't before? We saw that these T cells are expanding locally, and you can see marker for T cells CD3 in magenta and MHC class 2 in blue. So these T cells are expanding locally next to this also increase of MHC class 2. And which T cells are expanding? Both. T-cons, CD4 positive, OXP3 negative, and CD8s. Well, what about the B cells that I was mentioning before? We know that the Tregs here in green and B cells in white, they form clusters together. Follicular stages of B cells, characterized by the expression of IgD, drop once we remove regulatory T cells. And this is exactly the same effect that we see, and we haven't shown this in the paper, we have done experiments aside, with a TFR, a follicular regulatory cell, DTR mice. So when we remove these cells, also IgD positive cells drop. So here we are having the two effects, TH1 controlling the T cells, TFRs controlling the B cells. So all this is great, and I love immunology, and immunology by training and by passion, but immune cells in the meninges, they encompass around 30 to 40% of the total life cells. So let's do one step back, and let's do whole tissue RNA-seq. Let's take the whole tissue, lyse it, and see what are the main flavors changing in the meninges. So what we saw is that when we deplete regulatory T cells, there was an increase for interferon, especially interferon gamma pathways. And we went to validate this. And we see that T cells and a little bit NK cells were the major producers of interferon gamma, and they were increased when we remove regulatory T cells from the meninges. So the meninges gets inflamed, T cells increase, they start producing interferon gamma. What about the brain that is just below the meninges? So the brain gets inflamed, and the brain gets inflamed by the same cells that increase interferon gamma production, T cells and NK cells. Now, again, let's do one step back. The brain is super complex, different region, different function, different composition. So let's take four regions, and let's first analyze where these CD3, where these T cells are in the brain. To our surprise, already early on, we saw there was this accumulation of T cells in the hippocampus. And when we did by pathway analysis, we see that the hippocampus was the region in the brain with the highest enrichment for interferon gamma response. And this started indicating to us, to our surprise initially, that this is specifically to the hippocampus and not other regions. We verified that with activation of neurons and markers CFOS, and we also see a major changes in the hippocampus with other regions changing either really low or nothing. And when we did the sequencing, we also include the cerebellum in addition of these four regions. And for example, the cerebellum didn't change at all. So most of the changes was happening in the hippocampus, a localized response to the hippocampus. Some of the regions were not even noticing the depletion of meningeal Tregs. So what is the hippocampus and why we should care about the hippocampus? Well, the hippocampus is one of the two regions in the adult brain that has stem cells, neuronal stem cells. These in the hippocampus, they are generated in this red region called dentate gyrus, and they're in charge of memory and learning. The other region with the stem cells is here next to the ventricles, where they produce the cerebrospinal fluid. They're called ventricular subventricular zone, and they're in charge of olfaction. We checked that region too, and we didn't see any changes, neither in the stem cells, neither in the immune infiltration. So again, specific for the hippocampus. So let's interrogate the function of the hippocampus. And we see that when we remove meningeal Tregs, there was an impoverishment of neurogenesis or neurodevelopmental pathways and an enrichment of glia activation. We validate these by confocal microscopy, whole population RNA-seq, we sort them and sequence them, and single-cell RNA-seq of the hippocampus, this glia activation, especially astrocytes, microglia, and oligodendrocyte progenitor cells. But these cells support the stem cells, so the stem cells by pathway seem to be, their function seems to be downregulated. So is that happening? And I'm here putting just how the protocol that we are using, or we have been using most of the whole paper, and just to highlight how short we are checking on the changes. And what we saw at the beginning by confocal microscopy, and this was done by a collaboration with Jose Luis Trejo in Spain, and her PhD Elisa Pintado, we saw an increase of these stem cells, also known as radioglia-like cells, RGLs, characterized by the expression of SOX2 and GFAP, double positive. So we were like, oh, caramba, we have more, but the function is lower, but we have more because they are dying. There's no compensation on proliferation. We further confirmed that by a flow cytometry using a marker for cell death, an Exin5, and a marker for apoptosis, a form of cell death, Cleef-Caspase-3. And also the single cell corroborated all this. So 3 different ways, same conclusion. There's accumulation of these cells, but they were dead or dying, and there was no compensation by proliferation. So great. Does it matter functionally? So we did, as I mentioned before, hippocampus is in charge of memory, and we are doing like a really short protocol to avoid systemic repercussion or systemic contamination in our readouts. So let's do a model for short-term memory assessment, what is called Y maze. Basically, it's a maze in the shape of a Y. We block one of the arms, and we let the mice explore. Then we remove it, we keep it away for a certain time, and we put them back. We put the mice back, now opening this arm that was blocked. The mice are curious, and they will tend to explore places that are new to them. So if they have a good spatial memory, they will go to the new arm to be like, oh, what is this new thing? What is over here? If they don't have it, everything is new, so they will do 50-50. So 5 days post-DT, on the protocol that we have been using, we see that the mice that has been removed regulatory T cells from the meninges, they really don't know what is new, what is old, so they do 50-50. The ones who have the Tregs, they remember, so they go to the novel arm to explore it. To our surprise, this deficit persisted at least 8 weeks post-Treg depletion. So this depletion of Tregs with the death of the radioglia cells in the dental gyrus of the hippocampus leave a scar that persisted at least 8 to 10 weeks. That is the farthest that we have checked. How is this happening? Well, we saw increase of interferon gamma in the meninges, increased interferon gamma in the hippocampus, maybe interferon gamma. So to do so, we block interferon gamma with antibodies, but we did intraperitoneal injections. Why that? The brain at the steady state has a barrier, the blood-brain barrier, that doesn't allow, it's tightly regulate what comes in and out the brain and doesn't allow antibodies to cross it. So we want to see if it's a meninges-initiated event. We are taking the blood-brain barrier's advantage. We are injecting antibodies systemically, which will block in the meninges, but will not access the brain to block anything in the brain. So we block interferon gamma, we deplete our cells, and we keep blocking the interferon gamma through all our protocol. And what we saw is that we rescue the radioglia cell-like phenotype. Basically, they don't die when we block the interferon gamma. So this is the mechanism of cell death, but how are T regulatory cells in the meninges controlling interferon gamma producing cells? And we're going to go back, I'm sorry, just to point out that it's a specific interferon gamma effect on the radioglia cells. We did neurospheres, we incubate them with interferon gamma at different stages of formation, and we see that there is a direct cell death, dose-dependent manner of these neurospheres with interferon gamma. The more interferon gamma, the more death. And addition of interferon gamma at the beginning when you start forming the neurospheres prevent neurospheres formation. So it's a direct effect on the radioglia-like cells. So how are they controlling these interferon gamma producing cells that are starting activating in the meninges, in producing interferon gamma, and then go to the hippocampus? Well, we saw this CD25, and when we block it, we saw the same phenotype. We know that T cells and NK cells need IL-2 to get activated. And I checked for phospho-STAT5, is a downstream signaling of IL-2. And we see that T cells and NK cells have an increased phospho-STAT5 after Treg depletion. So then we did two experiments, a loss of function and a gain of function. Loss of function, we are artificially going to take this IL-2. We're going to do like by giving antibodies that take the IL-2 from the media. We will prevent this burst of IL-2, and we should prevent T cell activation and interferon gamma production and all the brain phenotype. The other one is the other side of the coin, a gain of function. We are going to overwhelm Treg capacity by a huge outburst of IL-2 targeting non-Tregs. And overwhelming these Tregs, we expect to see the same phenotype as removing the Tregs. For the loss of function, again, same concept as interferon gamma, intraperitoneal injection of anti-IL-2, as well as we deplete the Tregs and we keep this anti-IL-2. So always removing IL-2 from the environment and see how they respond. For the gain of function, we use a IL-2 complex. There is antibodies that you can bind to IL-2 and give it to the mice, and the S4B6 clone will bind to the IL-2 receptor beta or CD122. This preferential will bind to T-cons, NK cells, and CD8s and will induce activation of interferon gamma. But there is the same complex with another different antibodies, tiny different, that's called ES6. That will bind to CD25, this high affinity receptor of the Tregs, which is highly enriched on regulatory T cells. Indeed, we check, and in the meninges, even an inactivation, CD25 is not upregulated by T-cons or CD8. So by giving this antibody to this high affinity receptor to CD25, we can specifically deliver IL-2 to T regulatory cells. So let's go first for the loss of function. Here you have the protocol. Meninges, we see that when we prevent this IL-2 outburst, we prevent T cell expansion. We prevent T cell and NK cell infiltration in the brain, and we rescue the radioglia cell death. Now, the other side should be the opposite of this, right? If this mechanism is true. In yellow, the complex activating T-cons, NK cells, CD8s, and in blue, the YES6, the complex expanding Tregs, bringing IL-2 to the Tregs. When we overwhelm the Tregs, T cells and NK cells expand. With the only activating the Tregs, nothing happens as it should be. These cells enter inside the brain parenchyma, and they start killing radioglia cell-like through interferon gamma. So, in summary, we describe a new tissue Treg population in the meninges that has three flavors: YSN, TFR, and TH1. TFR controlling B cells, TH1 controlling T-cons, CD8, and NK cell activation. When they are present in the meninges, the hippocampus is happy, radioglia cells are fine, and the glia is in a homeostatic state. When we remove them, there is an excess of IL-2 that allows activation of T-cons, CD8s, and NK cells. That activation elicits interferon gamma production. These cells goes inside the brain, produce interferon gamma, activate the glia, and kill radioglia-like cells. So with that said, I just want to acknowledge and thank the lab, especially Diane, for trusting me in this new project that we were not familiarized, but she has been always extremely supportive with my crazy ideas and